Now, let us introduce you the part 3, Bioluminescence Imaging and Fluorescence Imaging. Let us first see bioluminescence imaging. Bioluminescence imaging, BLI, relies on detection of light from luciferase-expressing cells in an animal. This commonly achieved through implantation of cells engineered to express luciferase constitutively or by use of transgenic animals that express luciferase in one or more tissues of interest. Emission of light from these cells or tissues occurs following systemic injection of the luciferase substrate, luciferin. Bioluminescence imaging is a rapid, non-invasive imaging method that employs light-emitting reporters and external photon detections to follow biological processes in living animals in real time. Bioluminescence tomography, BLT, bioluminescence imaging is a non-contact optical imaging technique based on measurement of emitted light due to an internal source, which is then often directly related to cellular activity. It is widely used in preclinical small animal imaging studies to assess the progression of disease such as cancer, aiding in the development of new treatments and therapies. For many applications, the quantitative assessment of accurate cellular activity and spatial distribution is desirable as it would enable direct monitoring for prognostic evaluation. This requires quantitative spatial reserved measurements of bioluminescence source stress inside the animal to be obtained from BLI images. This is the goal of bioluminescence tomography, BLT, in which a model of light propagation through tissue is combined with an optimization algorithm to reconstruct a map of the underlying source distribution. To allow a more quantitative analysis from BLI, methods that allow the recovery of spatially reserved tomographic maps of bioluminescence source, location, and intensity have been proposed to allow bioluminescence tomography, BLT. The basic date is to employ a forward model of light propagation through tissue to the skin surface in conjunction with an optimization algorithm to reconstruct the underlying bioluminescence source distribution. For single wavelength state, this inverse problem is highly non-unique. Example, identical measurements are produced by many different light source distribution. However, as bioluminescence sources have broadband emission spectra, spectral reserved detection schemes can be used to measure the emission due to the bioluminescence activity at the surface of the tissue at several wavelengths in order to reduce the non-uniqueness. 3D modeling and reconstruction algorithm as applied to multi-wavelength 3D BLT image reconstruction have widely been developed. Fluorescence imaging. Fluorescence imaging is the visualization of fluorescence dyes or protons as labels for molecular processes or structures. It enables a wide range of experimental observation, including the location and dynamics of gene expression, protein expression, and molecular interactions in cells and tissues. Fluorescence sampling of cellular function is widely used in all aspects of biological, allowing the visualization of cellular and subcellular biological processes with spatial resolutions in the range from nanometers up to centimeters. Imaging of fluorescence in vivo has become the most commonly used radiological tool in all preclinical work. In the last decade, full-body preclinical imaging systems have emerged with a wide range of utilities and niche application areas. The range of fluorescence probes that can be excited in the visible to near-infrared part of the electromagnetic spectrum continues to expand, with the most value for in vivo use being beyond the 630 nanometers wavelengths. Because the absorption of light sharply decreases, whole body in vivo fluorescence imaging has not yet reached a state of neutrality that allows its routine use in the scope of large-scale preclinical studies. Fluorescence-mediated tomography, FMT, enables non-invasive assessment of the three-dimensional distribution of near-infrared fluorescence in mice. The combination with the microcomputed tomography, UCT, provides anatomic data, enabling improved fluorescence reconstruction and image analysis. Default and high sensitivity scans were acquired, and background signal was analyzed for 3 FMT channels. Analysis was performed for the original and an improved FMT reconstruction using the UCT data. Fluorescence-mediated tomography, FMT, is a promising imaging technology which allows three-dimensional assessment of the fluorescence distribution in whole mouse bodies. FMT is often used with contrast agents, example, fluorescence probes, which target tissues or receptors of interest. In addition, activatable probes can be used to assess the presence of enzymes, or the fluorescence may originate from reporter genes causing expression of fluorescence proteins. Another typical application of FMT is the determination of biodistribution of novel pharmaceutical drugs and drug delivery systems. Okay, that is all for this part. Thank you.